Hi, I'm Marloes Peters, and I have been working with molecularly imprinted polymers since my PhD at Hasselt University in Belgium. So, molecular imprinting, I have like a short video where I talk about the introduction to this concept, but in essence, it is you start with something that you want to detect or like a compound that you're interested in, you assemble some monomers around it, you polymerize it, and then you extract it. So, you end up with a very porous material which should be able to bind whatever target you're interested in with an affinity just as high as antibodies, but having the advantages of low cost, robustness, and being able to tailor it as you want. So very traditionally, the main concept of molecular imprinting or the commercial applications have been found in purification and extraction, because there you can quite simply work with microparticles. So the structure wasn't that important, and these microparticles you can quite easily produce on a big scale. But in recent years, you can find that there's like an upcoming of other applications, some more biomedical applications, and this is what I'm going to explore in this video, where I'm going to focus on drug delivery. So how should this work? Well, imagine like an antibody can anchor onto a cell onto a very specific receptor. So these antibodies can work as therapeutic agents as well. Now imagine if you have this synthetic mimic, this molecularly imprinted polymer. Similarly, it can bind to the exterior of cell surface if you imprint with the protein or an epitope of the protein, which is on the outside of the cell surface. So that's similar to what you can kind of see in that image here. You can see the nanoMIP, which kind of looks like a little Pac-Man, which really specifically binds to the exterior of the cell. So we don't really know so much yet, and I'll come back to that later, whether these actually go into the cell. But for now, let's say we focus on proteins or structures which are on the exterior of cell walls. So there's a couple of different approaches you can take. And let's just focus on the first approach for now, where we look at a so-called double imprinting approach, or using a dual functionality of the MIPs. In general, if we look at nanoparticles, because we want to look at biocompatible systems where we need to go to this nano format rather than micro. And what we can do here is that we can imprint with the protein of interest. And then we can also do like a second imprinting step where we kind of load in the drug. And I will show in the next slide what this looks like. But in essence, the drug is not bound by very strong non-covalent interactions, unlike the receptor. So it means you will get some slow drug delivery over time. But you can do that, but you can also use other dual type of functionalities. So you can find in literature examples of people incorporating, for instance, certain enzymes onto it. And so these enzymes, for instance, might cut off sugars. So if you look at like a therapeutic type of effect, you can imagine that what this does, the nanoMIP, and this is particularly used in cancer therapy, can specifically bind to specific cells, and when it gets there, you can trigger like a second functionality, which can be either delivery of a drug or something else that might enhance the therapeutic effect. The second approach is that we can look at stimuli-responsive molecularly imprinted polymers. So that can be based on pH, which is a very common approach. It can be based on temperature, so using very specific monomers that are temperature responsive. But you can also look at, for instance, specific sulfur groups, a glutathione, so you can look at kind of redox molecules that you can incorporate. The final approach is we can embed these MIPs into porous structure. So you can use, for instance, certain type of hydrogels and porous matrix. And when slowly the MIPs kind of come out of that matrix, that's another way of how we can control the drug delivery. Now, let's look at the double imprinting approach. So here, as I said, you can see this is very standard to traditional solid phase imprinting. And in traditional solid phase imprinting, you use a glass bead as an affinity medium, and you really functionalize your proteins onto that specific glass bead. Now that's similar here, and then you polymerize around it, but what you do differently is that you polymerize in the presence of the kind of target that you're interested in. So that's why you end up with like a nanoparticle where you both have the imprinting of like a protein or an epitope of interest, and then to a lesser extent, and this is why it facilitates drug delivery, a certain therapeutic. I think you can imagine, if you look at that approach, like what kind of problems that might give, because obviously normally MIPs are very stable. This is not the case with this system because you get very slow drug delivery. You would always see some leaching of the drug out over time. So the stability is a problem. We're also looking at two very different targets which might work under very different conditions. So you need to make sure that the monomers work for both. But also then the solubility can be a problem because lots of these kind of drugs that you look at are very hydrophobic, so they don't necessarily dissolve in water, and we tend to do our nano-MIP synthesis in water-based systems. So very complicated, and that's why you can't really find much about this approach yet, I think. So I already mentioned like some challenges that you would have, and let's presume that we're working with these nanoparticles. And let's just go over some of the complications that you might encounter. And I would say that the first point is directly, I guess, the most pertinent one. The biocompatibility is so important. So you can find examples of nano-MIPs that have been injected into mice where they might claim this is biocompatible, but really much more research is needed into figuring out whether that's really the case. And most of the cases when people kind of present this in literature, they talk about that the functional monomers are biocompatible, but really the majority of the compound in most of the cases is actually the cross-linker monomer. So it's not just that, you need to consider the whole system. So even the initiator that you're using, your cross-linker monomers, also making sure that there's no kind of leaching out of the solvent, and that can be very complex. The other part is that the size really matters. And tailoring the size, and I'll come back to that, is really not as simple as you might think. The second point relates to the selectivity. So how do we really know that we capture like the right receptor onto a specific protein, onto a specific cell? And it is known that imprinting of proteins is far more challenging than imprinting of small molecules, where you know where your binding sites are. Whereas a protein, you might have an infinite number of binding sites. So it becomes much more complex to think about what you want to target. And then even more so, if you look at, for instance, cancer cells, often it's not a case of the receptor there or the receptor is not there. You look at overexpressed systems. So how can you make sure that it only goes towards the cancer cells if you also have a small portion of receptors onto a normal healthy cell? And I guess that's part of the reasons why a lot of these drugs, chemotherapeutic drugs, encounter a lot of side effects. The third point relates to the drug delivery rate, because tailoring that is not an easy task in whatever kind of commercial system that you've got. And particularly if we look at this double imprinting, we look at very minor non-covalent interactions. So how do you know that's really sufficient in order to make sure that you can facilitate the exact drug delivery rate that you want? And then imagine if you do it from a porous matrix, that might even be more complicated. The final point to consider, and this kind of, I guess this hangs into the biocompatibility, we don't really know so much yet what these nano-MIPs actually do to the cells. So in a way, would they actually enter the cells, or do they just stick to the exterior? And there's really evidence to suggest that they actually do migrate into the cells, but there's much more work needed to kind of figure out how that happens and how it's being cleared from the body. And things to consider there is that it really depends on the size. So the particle size needs to be 100% uniform, which you might be able to do, but often then you compromise your yield. And the system needs to be really reproducible. So you need to start looking at maybe some controlled polymerization strategies. So here I'm going to go over two examples. The first one is not so much drug delivery, but it's more MIPs for like therapeutic applications. And in this case, it's looking at immunocapture of circulating tumor cells. So it uses kind of like a dual approach where they use gold nanorods, which exhibit a photothermal effect. And then they have a hydrogel, which is imprinted with cells in order to kind of capture these circulating tumor cells. So later on, like if you find later publications, they also combine this with magnetic beads. So this is really a surface. So you can imagine you would kind of take out some blood and you can capture circulating tumor cells. So it's a way of you would be able to purify it. But you can imagine that you can transform this into a nanoparticle as well. And these nanoparticles, when injected, should be able to kind of capture out these circulating tumor cells. Now, if you wanted to do this kind of like in like outside of the body, so in vitro. They've also combined this with magnetic beads, so that should be able to far easier collect your nanoparticles. And you see this quite often in purification cartridges, where rather than having like a standard like glass bead as a core, that they work with some magnetic nanoparticles, which could be very much of interest. And what I liked about this paper, that it's specifically triggered by an NAIR response, so using that kind of stimuli responsive effect where you can kind of switch things on and switch things off. And this way you can capture and you can tailor the capture and the release of certain drugs and cells. Now, in the first example, you could see that there was like a response towards a change in temperature. And this one actually looks at a response in pH, so that's somewhat different. And here you will see they target another very common marker, which is overexpressed in a lot of cancers, and this is sialic acid. So again, this is overexpressed. It's also present in normal cells. Now, what's different here is that they didn't include the actual drug itself, but they looked at a prodrug delivery system. And remember that, like, if you know the term prodrug, a lot of drugs don't, for instance, are not the actual drug compound itself because they're too unstable, or they would be metabolized far too quickly before they come to the point of action, particularly if we look at like oral tablets. So the prodrug is actually can metabolize into the actual drug in the body and then have the effects. And that has a lot of advantages because the prodrug is probably a lot more stable than the actual drug. So overall that can enhance the efficacy of the system. Now what they exploited here, so before we had like an NIR response to light. This one actually looked at specific changes that you might get in the tumor, because as we know, the tumor microenvironment tends to be somewhat more acidic. And we're not looking at big changes in pH here, so our normal blood is 7.4, but we know that here they said it's around like 6.8, 6.9, and they use that to kind of justify that the particles that they have would only really go or migrate towards the tumor cells and there deliver the drug of interest. And this was actually tested into mice because the first thing they want to look at is the clearance in the body, and they could see that this was nicely cleared by the liver, which is what you want. What you don't want is that these particles end up in fatty tissue and that they accumulate here. Now, what they've also seen is, unfortunately, they do have to sacrifice the mice for this. What you can see that when they looked at the tumors, that the majority of these molecularly imprinted nanoparticles did end up at the site of the tumor. So that means that it should, in theory, increase the drug efficacy. But as you do say, like in general, the tumor microenvironment is acidic, but obviously this might not apply to all specific systems and work better in certain cancers compared to others. Now, in general, I really hope that in the next couple of years, we're going to see a lot more of MIPs in biomedical applications because there's nothing really on the market yet. And there's nothing that's really been approved and thoroughly tested into humans. So real advantages would be the low costs and the robustness, and specifically being able to tailor your MIPs to whatever that you're interested in. So sometimes exploiting these dual functionalities. So it gives you a lot more options compared to normal antibodies, for instance. However, I think a lot more research is needed for the biocompatibility, and as I said, considering like the whole system, and we need to see far more controlled polymerization strategies because in general MIPs are still produced using free radical polymerization, and that doesn't really allow you so well to control the size of the particles. We also need to look at more scalable strategies because the yield, especially when you look at solid phase imprinting, is very low. So really we need like automated flow reactors to make sure that we can specifically scale this up. The selectivity is a particular problem, especially when you look at cancer therapy, where I guess the majority of these drug delivery systems are based on for now, because if you work with sensors, you can use a non-imprinted polymer as a control. So it's very easy to embed a control into your system and you just compare the two, and the difference between the two is a specific binding. However, in terms of the drug delivery, you obviously can't have that, and that might lead to side effects. So the selectivity is far more of a problem, I would say, than in kind of more traditional MIP applications. And then the final point to consider, so we know that antibodies also act as therapeutics, for instance, in immunotherapy. So in the future, who knows, when we know more about how these MIP bind to the cells, like, would it be of interest to also start using them as therapeutics rather than just for drug delivery? So thanks for watching, and do have a look at this playlist where you can also look at, for instance, how we've exploited molecular imprinted polymers for the very specific detection of COVID-19.